Thank you very much for inviting me today to talk about novel bioassays in Picodroplets for single-cell analysis. I am Romina Durigon, and I am working as a senior field application scientist for Sphere Fluidics. I joined the company 7 months ago, and before that I was working in the lab as a scientist. I investigated the role of mitochondrial dysfunctions and metabolism in rare diseases, especially mitochondrial rare diseases, but also in cancer and aging. Thank you very much. It's a pleasure for me today to talk about Picodroplet technology for single-cell analysis. The single cell is the smallest functional unit of living beings, and bioengineers and biologists have long studied life at single-cell level. The first single-cell study was published in 1958, so more than 70 years ago, by who became later a famous immunologist, Gus Nossal, and a Nobel Prize winner, Joshua Lederberg. Their study was instrumental to prove the clonal selection theory was true. That was to say that the 1B cell is only activated by one antigen and produces antibodies of only one type. To show that theory was true, they used a very elegant and laborious method, which included the immunizations of rats from antigens of 2 Salmonella strains, and then the isolation of plasma cells, and they used the spray cell suspension method to encapsulate single cells into mineral oil droplets. By doing this, they were able to generate hundreds of droplets of various sizes, as you can see here from the picture on the right. They analyzed 456 single cells and reported that at the single cell level, none of the antibody-producing cells had signs of dual reactivity. The study was the first of many studies to come at single cell level. Microdroplet-based technology is a consequence of the miniaturization process that was envisaged a year later in the famous lecture given by the physicist Richard Feynman in 1959 at the California Institute of Technology, called "There is Plenty of Room at the Bottom." But what is microfluidics? Microfluidics is a science of fluid dynamics on the microscale, and is based on concepts borrowed from physics, chemistry, biology, engineering fields. And droplet-based microfluidics is an essential branch of microfluidic chip research that forces two immiscible liquids through channels, leading to the formation of plugs of one fluid within another fluid. And when talking about picodroplet, we refer to the generation of droplets on the picoliter size. To generate droplets, two immiscible liquids are necessary. One is called the continuous phase and covers the droplets, and the other one is called the dispersed phase or droplet phase. According to these definitions, droplets can mainly be divided into two categories: water in oil type, in which the continuous phase is the oil and the dispersed phase is the water, or oil-water type, or oil in water droplets. Droplet generation also typically is either passive or active, depending on the need for external forces. Passive flow does not depend on external energy and relies mainly on the geometry of the device that is used for the droplet formation, whereas the active method includes electric, magnetic, and other type of forces. On the left two videos, we show a type of passive flow generation for droplets, which is called flow focusing, to generate water in oil droplets. Water in oil droplets are formed when the solution with the salts and the reagents come in contact with the oil that comes from the top and the bottom. And when the two liquids meet, the shear forces of the oil generate droplets. The top and the bottom video, they show the single cell mode, so single cell encapsulation within the droplet, or multiple cell mode, multiple or mini pool of cells encapsulated within the droplet. Microdroplet biochips comes also with various unit operations to manipulate droplets content either actively or passively. For example, merging or fusion of droplets can be done either actively or passively. In an active mode, an electric field is applied to induce instability across the interface of droplets, which results in fusion of the droplet. The passive approach for fusion involves the exploitation of channel geometries to achieve the fusion of the droplet. For example, the device configuration can slow down the droplet flow, allowing them to come closer together and fuse. Another type of picodroplet manipulation is called sorting, which can be done based on the size or the content of the droplet, or splitting or fusion that in passive modes, for example, or in active mode. In passive modes of droplet fission, T-junctions and other means of obstructions are introduced in the microfluidic chip, leading to the splitting of the droplet. Active fission, instead, approaches this by using an electric field. Dispensing is another type of manipulating the droplets, which can be single dispensive droplet or pool dispensing, when you dispense a pool of droplets together. And another type of manipulations is the mixing, when you mix droplet content together. But why are picodroplets becoming an attractive tool for biomedical applications? Since its advent, droplet-based microfluidic systems, also here referred as picodroplets, have been extended to several biological and chemical applications. Now we see why. One of the main reasons is because the ability to produce picodroplets at high frequency enables high throughput biological screening and analysis, which would not be possible with the conventional standard method. Also, each droplet is monodisperse or highly uniform in size, which means that it acts as a miniaturized and independent experiment. The confinement of cells in the picodroplet environment ensures that any material, any molecule that is released from the cells is not lost, but is concentrated in a picoliter volume, making the detection method faster. In addition, the ease of manipulation of droplets further establishes the applicability of picodroplet technology for various cell-based assays. In this slide, we see that microfluidic technologies has various applications in disciplines that can go from drug discovery, drug screening, antibody screening, single-cell analysis, cell line development, cell engineering, cell therapy, including CAR T-cell therapy, but also medical diagnostic and tissue engineering. Moreover, in the field of synthetic biology, picodroplet microfluidic technology has shown exceptional promises for synthetic biology experiment, including DNA assembly, transformation, transfection, culturing, cell sorting, and plays also a significant role in food safety, sensing, and several other applications. Today, we will focus on the advantages and the use of picodroplet microfluidics in antibody discovery. What are the advantages of using picodroplet microfluidics for antibody discovery? By looking at the number in this table, we see that on average, a single cell secretes 2,000 molecules per second, and in 4 hours, the concentration of the secreted molecule would become 50,000 times higher than the concentrations of the secreted molecule in a 96 multititer plates well. This means that screening single cells in a picoliter range volume is faster than a conventional method, which would take days before becoming detectable. In addition to speed, the significant reduction in volume facilitates an equivalent reduction in the background and a drastic increase in the local concentration of the molecule of interest. And also, by encapsulating single cells, even or biomolecules in the picodroplet, the assay can be highly specific. At Sphere Fluidics, we have developed various novel bioassays in picodroplets that can be used for rapid and high throughput screening of millions of antibody secreting cells, here abbreviated as ASC. These assays use a fluorescent readout to detect the antibody binding activity inside the picodroplet. For example, on the left, we saw a FRET-based assay, which is a high-throughput screening of antibody-secreting cells using a Foster resonance electron transfer, or FRET-based detection assay, which I will explain on the following slide better. In the middle model, the middle model shows another high-throughput approach for screening antibody-secreting cells by co-encapsulating a single antibody-secreting cells with the target cells, or bits, that are expressing or exposing the antigen of interest on the surface, respectively. If the antibodies released by the antibody-secreting cells bind to the antigen on the co-encapsulated cells or bits, the binding can be detected by a concentration of fluorescent signal on the surface of the body. The right model shows a third way of screening antibody-secreting cells by co-encapsulation of a single antibody-secreting cells with the readout cells. This is to say, cells that will express a fluorescent protein, for example, in this case, green fluorescent protein, upon binding of the released antibodies on the antigen that is expressed on the cell surface. Now, microfluidics for antibody discovery originated in academics lab and is now a routine antibody discovery platform in pharmaceutical research and development labs. The antibody discovery workflow shown in this slide is taken from a 2021 paper published by the research and development Merck scientists. They use a Sphere Fluidics Picodroplet device called Cytomine to rapidly identify natively paired antibodies from millions of plasma cells, isolated directly either from immunized mice or vaccinated humans, in as little as 2 days. Indeed, the remaining steps of the antibody hit discovery workflow that include full antibody gene PCR recovery and subcloning for secondary screenings were completed within 4-6 weeks. So the advantages of using Picodrop technologies device like the Cytomine include speed, which is very important when we're working with primary cells, which are very sensitive, high throughput, again, very important when investigated IgG or antibodies repertoires for primary cells, and the possibility of running direct screens in the Picodroplet, for example, for selectivity, cellular binding, or cellular function at single cell level, as we will show in the next few slides. One of the Picodroplet assays used in the paper is called antigen-specific FRET-based assay. This assay enables the screening and sorting of single cells secreting antibodies against an antigen of interest. So the model shows that one antibody secreting cells is encapsulated within the Picodroplet along with the antigen conjugating to a fluorescent labeled FRET donor, here shown in green, and the fluorescent acceptor FRET probe, here shown in red. The binding of the secreted antibodies to the conjugated antigen and acceptor probes will generate a ternary complex. This complex will result in a FRET signal upon excitation of the donor fluorophore. Therefore, it becomes detectable. In fact, the scatter plot shown on the right shows the donor and the acceptor fluorescent signal recorded in the droplets during the sorting process via FRET. So empty droplets or droplets containing cells that are not secreting any antibodies will sit at the bottom right of the scatter plot, as their FRET signal is at the background level. Droplets, instead showing distinguishable and strong FRET signal, are those sitting at the top left of the scatter plot, as for them there is a drastic decrease in green donor fluorescence and similarly a dramatic increase in red acceptor fluorescence, and droplets with average FRET signal seen in between the high and the low droplets. So this is a results that the paper show when scientists use the antigen-specific FRET assay in fresh antibody secreting cells from human. On the left are the schematics of the workflow for sorting freshly human plasma cells for secreted antibodies specific to TT or tetanus toxoid via FRET. So plasma cells were isolated from vaccinated volunteers. Cells were then injected to the cytomine for single-cell encapsulation with tetanus toxoid-specific FRET-based reagents, and then the droplets were sorted and the hits were collected for downstream analysis. On the right is the relative average donor and acceptor fluorescence signals recorded during the antigen-specific sorting process. The positive hits or droplets are selected within the gate that includes 0.05% of the total number of the droplets that were generated with this experiment. Another Picodroplet based assay that used in the paper is called cell surface antigen binding assay. This assay enables the screening and the sorting of single-cell secreting antibodies that bind to the surface of a target cell, could be a tumor cells, for example. This requires co-encapsulation of one antibody-secreting cells and one target cells, along with secondary antibody. This in case is IgG-AF488 antibodies, as shown in the model on the left. Now, the graph on the right explains how the signal, fluorescence signal, look like when we have co-encapsulation different cell type, like in this case. The sorting of positive droplets is achieved by detecting the red channel for antibody-secreting cells that in this case were pre-stained with a red dye in combination then with green fluorescence for detection of target cells that will be decorated with antigen-specific antibodies detected by the green fluorescence signal given by the anti-488 mouse antibody. These are the results that the scientists obtained by running this experiment. So on the left are the schematics of the proof of concept workflow for screening for cellular binding. In particular, in this case, in these proof of concept studies, they spike two hybridoma cell lines. One is the anti-EGFR specific MAB108 hybridoma cell line, which produces antibodies against EGFR, and this was spiked 1:100 into a relevant hybridoma, which they call OKT3. The two hybridoma cells were co-encapsulated with target cells, which in this case is EGFR positive cells, which are cells that will express EGFR on their surface. The three cell population were injected to the cytomine for enrichment by screening for cellular binding by the green channel and followed by the downstream analysis. On the right is the signal recorded during the sorting process by plotting the target cell signal in green on the y-axis and the antibody secreting cell signal on the y-axis. From the gated population of droplets, droplets containing only red signal or droplets containing only green signal were excluded, so that droplets within the gate are the positive hits, and that those droplets were selected for being dispensed and analyzed by PCR. A total of 288 droplets were dispensed and 241 were analyzed by PCR. 77 of these droplets contain G-specific for MAB108 hybridoma, which was what they wanted to enrich for. This indicates a successful enrichment from 0.3% from the initial cell population to 32%, which was a remarkable result. So summarizing, what are the advantages of Picodroplets for antibody discovery? First of all, single-cell encapsulation is a unique platform for capturing and quantifying secreted antibodies in a few hours, compared to standard assay where it would take 2-3 days. By confining single cell in a picoliter environment, droplet microfluidics enables studying the specificity of the single cell. And because all droplets receive the same amount of reagents and a threat reaction occurs only in the presence of the secreted antibody, the threat process ensures high sensitivity. The platform also enables high-throughput single-cell phenotypic screening, for example, in this case of IgG antibody secreting primary cells, or large recombinant antibody secreting cells libraries for both soluble and membrane-bound antigens. In this case, the genotype-phenotype linkage is not lost, unlike conventional high-throughput screening of single cells, like, for example, the flow cytometry, that are restricted to intracellular or membrane-bound epitopes. I want to thank you very much for your attention, and I'm very happy to take any questions, and please do not hesitate to contact me if you need more info. These are my details. Thank you very much again.